Est-ce que tu m'entends? Ça va quand je parle?
Bon, ben bonjour, bonjour à toutes et bonjour à tous. Aujourd'hui, nous avons le plaisir d'accueillir Yoël Forterre qui vient avec le soleil marseillais. C'est super. Donc Yoël, je sais pas, je pense que tout le monde connaît Yoël, mais c'est toujours un plaisir de le voir et de l'écouter. Il va nous parler, donc c'est spécialiste de pas de matière molle en général, mais de mécanique ou de mécanique molle, mais de granulaire, de plantes. Et il va nous expliquer, je pense, encore tout ça aujourd'hui. À toi, Yoël.
Okay. Thank you very much, Pierre-Yves. Thank you everyone for being here. Good morning. So yes, I'm going to talk maybe about a topic you are not necessarily very familiar here in d'Alembert. I'm going to talk about plant biomechanics. So this is a work done by many people in Marseille, so I will present them along the slide. And more precisely, I'm going to talk about mechanics of rapid motion in plants. All right, so I'm going to talk about plants and to talk about movements. And usually it's not necessarily the first things that comes into the mind when we are thinking about plants, but actually plants are moving all the time, obviously, as any living organisms. And here there are two pictures. On the left, what you see is a close-up on the surface of the leaves and at the cellular level. And what you see, those little cells, actually you see two cells making a kind of a peanut. And actually those cells are called the stomata, and they are responsible for the exchange of gas between plants and the atmosphere. So they are critical for plants and for everyone because they are responsible for the photosynthesis by absorbing carbon dioxide and releasing oxygen. And those valves actually open and close slowly depending on the day, depending on the environment conditions. So this is a movement at a very small scale and very important one. And on the right, I put here a completely different scale. So we are here talking about tens of meters, and those are trees. And what is surprising in this picture is that you see that those trees seems to grow in a wavy mode, all right? And the reason for that is that actually when the tree were very young, they have been laid down on the ground when they were short. And during the growth, they detect gravity, so the vertical direction. And instead of growing isotropically around the trunk, one side of the trunk is growing faster than the other, and you have this bending motion, okay, which is completely irreversible, of course. So this motion is reversible. This is swelling and shrinking of the cell, and this is growth, so plastic deformation. And so plant moves at all scale, but what we know also is most of the time plant motions are pretty slow on our time scale. So here also I have two examples. So two accelerated movie. Here 1 second equal 1 hour. Here the 1 second equal 100 minutes. And here this is on the left. this is a very classical pine cone cut in two and put in an atmosphere where the humidity is periodically changed, all right? And here it's, again, a grass, a little plant called Arabidopsis thaliana, so it's a kind of a Drosophila for plant biologists, which is suddenly put at 90 degrees versus the vertical, and that responds to gravity as the tree I've just shown before. And so if I launch the movie, so what you see here is the fast and slow reopening of the cone. And Etienne knows this very well because he worked on this some time ago. So it's a purely passive motion driven by humidity gradient, whereas here it's an active response, growth response to gravity. And there are actually gravity sensors inside the plants. Okay, so most of plant movements are slow, but some are pretty fast. And these are the ones I'm going to talk today. So I'm going to talk about this plant. This is the sensitive plants, Mimosa pudica, and this is a real-time movie this time, so no acceleration. And someone is bringing a lighter here, so heating stress. And if I run the movie, what's going on is that you see both a very fast folding of the leaves, and what is also amazing, you see a very fast propagation of the signals that you have imposed at one end of the leaves, all right? So we are really interested in this kind of thing. And another movie where this time, even with your naked eyes, you cannot see the motion, so rapid it is. So here you need fast camera, and that's a pretty old now documentary from the BBC, where there are not tens, but many very nice movies showing very fast plant movements, especially movements whose functions is for reproduction, so to spread seeds or pollens rapidly at large distance. And for these plants as well, recently there have been a nice paper by the Manchester group, Finbox et al. In PNAS, about how this skirting cucumber ejects his seed. All right? So plant moves at all scale and over a very wide range of time scale, but obviously, unlike us, they do not have muscles, so that's for sure. They have actin, they have mucin, but what is very peculiar to a plant cell compared to animal cells is that if I sketch very roughly a cell by balloon, what is different for plant cells than animal cells is that the plant cell is surrounded by very stiff and elastic materials called the cell wall, mostly made in cellulose. Okay, so it's a kind of a gel, polymer matrix, highly hydrated, and that with a Young modulus about gigapascal, so it's pretty stiff. And thanks to this stiff cell wall that surround plant cells, but that surround also fungi cells, that surround also bacteria cells. So many organisms are walled organisms. Thanks to this wall, plant cells can accumulate solute and accumulate osmotic pressure and hydrostatic pressure, actually. So by having a solute mismatch between the inside of the cell and the outside of the cells, because of the cellular membrane, which is here in dotted orange, let's say, because this is a semi-permeable membrane that blocks the solute but let freely the water go through, you have a water potential difference that appears, a chemical potential difference. And so by osmotic, the cell is absorbing water until the pressure is high enough that there is a thermodynamic equilibrium between the inside and the outside, right? So it's a kind of a stretch balloon, a plant cell. And the standard view of how plants can produce motion, is usually that it's due to water flow, at the cellular level at least, and due to osmotic or cell wall change, you have change of cell volume and then motions. So that's the basic view if we look at what I call the motor unit of the plants, which is the cell, this osmotic cell, all right, with the cell wall. So the question now we are asking now is, okay, this is for one cell, but now if you want to produce an efficient motion at an organism scale, then you have to upscale these mechanisms. and how plants manage to do this. So how to upscale cell motions to tissue and organ levels, that's a very basic questions. And also, I would like in this talk to focus mostly on what I call active motion, not passive one. So I've shown the pine cone, for instance. It's a purely dead tissues that swell and shrink due to change of humidity, but there is no biological process in this motion. The two motions I'm going to talk today involve active motion, which means that they are tightly related to some biological process. And it's interesting to study them as well, because it can put some limits to or question some biological process as well, not only physical or engineering questions. All right? So as I said, I will focus on two plant motions today. So the first one is the Venus flytraps, which is a carnivorous plant. And what I'm going to show is mainly the work of during the postdoc of Jong-eun Ryu, who is she's a South Korean postdoc, now come back in South Korea. And the second part will be on the Mimosa pudica, so the plants I've just shown. And this is the postdoc of Mathieu Rivière, who is currently in the lab. All right, so let's start with this one. And this one, we started to work on it a long time ago. Actually, this is how I go into this field, by starting to work on that during my postdoc long time ago. And so the Venus flytrap was originally, scientifically, I would say, first studied by Darwin, who called it the most wonderful plant in the world, because it's pretty amazing. So you see the trap of this plant, and the trap is inside the trap you can see some trigger hairs, and when an insect touches the trigger hairs, the trap is triggered. And so this is again a real-time movie of the motion. And so long time ago, when so just before, what is known is that when you touch the hair, what happens is that you trigger an electrical signals. So that's pretty amazing, and that's what amazed the most Darwin at that time. I mean, seeing this rapid motion, he suspects that there should be some electrical signals, and so he suspects that there should be some nerves, some muscle, and he asks some physiologist friends to make some tests with electrode to see whether there is electric signals. And indeed, there are. So people who measured on muscle frog electrical signals also at that time measured electrical signals in plants. So there is this spike that propagates much slower than how nerves, how the action potential in our nerves. It's about 10 centimeters per second. And then very recently, what it has been observed is that after this electrical signal, there is a molecular signaling. And what you are going to see actually here is a movie from a group in Japan, where they can tag the calcium that goes inside the cells. So when it goes bright, it means that some calcium goes inside the cells. And I run the movie. Sorry, it seems that, yeah. You see, I run the movie once, and then there is first touch, and then second touch. You see that very rapidly there is a kind of calcium waves that propagate all over the traps, and that trigger the traps. So this is the, let's say, the signaling pathway part, and I'm going to focus on the mechanics more. So what's going on after these signals? Yeah. No, it's because actually there is a continuum of cytoplasms between cells. So cells are tightly packed, and they communicate by a tiny, let's say, bridge or hole. And actually, the cell membrane itself is a continuum. I mean, it's not a closed pocket. It's a continuum along all the cells, which means that there can be a voltage difference between the inside of the cells and the outside of the cells all along the cells. And then this is a bit like the mechanisms of action potential transmissions. So it's a local depolarization of the membrane that then spatially propagate as an excitable system. So there is really a critical voltage, and then above this critical voltage, then you get an instability. But it's still not fully clear. Yeah.
Just the concentration concentration of the ion or?
Yeah, ions, yeah, mostly potassium, chlorides, and also sodium. Yeah. And yeah, so initially I start to say that I worked on this long time ago with Jan Skottheim, Jacques Dumais, and Mahadevan, where we first focus on the purely macroscopic aspects on this motion. And what we show is that actually this motion, this rapid closure of the traps, has some things to do with the geometry of the trap. And you can reconstruct the shape of this trap, and it's pretty clear that actually it's like a thin shell that is curved outward initially, and then that is curved inward at the end when it's closed. And here, this is the mean curvature average over the whole trap of the leaves. And you see that when you trigger the traps, nothing happens. Then there is a kind of a slow part, and then a sudden snap where the mean curvature completely reverse sign. And this is also correlated. This is not shown here to the change to the Gaussian curvature dynamics, which goes through a minimum when just before the snapping. All right. So that's for sure. There are some purely passive macroscopic. Yes. This is an observation. So actually, this was obtained by stereoscopic reconstruction of the shape. So with high-speed camera. So it's a 3D experimental shape as a function of time. And then from that shape, since we can, using finite difference methods, but from the experiment, we can reconstruct the geometry and then differentiate it. Yeah, it's just you know, it's very easy. The plants is all right. You can put fluorescent one. You will see later. No, it's all. You do this gently. Yeah, gently. If you yeah, there are almost no sensors outside. All right, all the sensors are inside. It's true that if you poke too much the outside, then it will close. But if you do this gently, it's all right. Yeah. So okay. So there is this obvious analogy between disclosure and the snapping of a shell. Here I just write the simplest elastic energy you can write by combining the bending elastic energy and the stretching elastic energy. And the stretching elastic energy, of course, in this very simple sketch, has two well and an elastic energy barrier. And this is why going from that state to the other states, you have to overcome an elastic energy barrier, and then you accumulate your stored elastic energy that are suddenly released. Okay, that's the basics of any snap buckling instability. But what we also all know about snap buckling instability, it's usually, or not always, but some of the states is metastable, which means that if you are stuck there, you will have no motion, spontaneous. All right, and actually this is a toy that this is the toy you all know, that the snapping shell. I reverse it, nothing happens, right? It's in a metastable trough, and in order to snap it, I have to apply, for instance, an external force, and then it snaps, right? So the question is, what is this motor for the plants? This is the question we have tried to ask. Okay, what is the internal mechanisms that the plants use to actively change, for instance, its intrinsic curvature in order to go through the elastic energy barrier of the stretching elastic energy? I mean, what is this motors? What's the mechanics of this? What's the time scale? And we want, so that's a more difficult part than just, you know, measuring the shape and say, okay, there is this analogy. And because now you have to, to go inside the organism itself. I mean, the cell. Yeah. Yeah.
But is there stretching as well?
Yeah, this is the stretching energy, and this is just a toy model of. This is not the full elastic energy of a shell, but this would represent the stretching energy, which means that this is, let's say, this is the Gaussian curvature. So you have stretching energy if you change the Gaussian curvature compared to the natural Gaussian curvature, and this is the bending energy. Okay, so you have a terms to the power force for the curvature, and here a terms to the power 2. Okay, so this sketch corresponds to the stretching energy here. Yeah. So this is how the project starts with Jong'un. We say how to reveal this active dynamics, how to suppress the purely passive part of the motion in order to reveal what's going on internally. And the idea was very naive initially. We say, okay, let's remove the shell. Let's remove the geometry. And by doing this, you just slice and you transform your shell in a series of beams like that. It's very intrusive, I agree, but we tried this. As soon as you cut a trap, it closes in this case. And it's not like putting dots on it. It really closes it. But then, so you suppress this. You hope to suppress the stretching elastic energy terms, and then you only see the natural curvature change of the beam. So just two movies. So this is a movie of an intact trap. So in this case, this is fluorescent dot, and this is real-time movie. So I run it, and you see rapid snap again in about 1/10 of a second. But now this is what I call a cut trap. So you see here, as soon as I've cut the trap, it closes, but then it reopens in about 24 hours. Right? And then now I have a reset trap with no longer shell. And I can trigger it. So it won't be as dramatic as before, of course. So just look carefully. I run it. So Jong'un triggered the trap. And this is what you see. So this is real time. So you see a slight change, but still there is a change of natural curvature over a time scale which is about a second. So it's not as fast. Maybe it's too slow to catch a fly, but it's still a rapid motion for a large body of tissue. All right, and so you can compute not compute, but measure things. And what you see is that as soon as you have cut trap, you decrease strongly the maximal closure velocity by about an order of magnitude. So incidentally, this is a kind of late proof, let's say, that actually the snap buckling instability matter. I mean, the fact that it's a shell is important to reach such high speed. But still, so this is the dynamics of the active bending here. And you see it's a nice exponential dynamics. And the time scale is about a second, okay? And the deformation of this beam is about few %, all right? From convex to concave. So it's slower, but it's still fast. So now, of course, in order I mean, you expect referee to read this and so on. So, of course, we try also to extract this active dynamics, not only with this technique, which is highly intrusive, but also with another one. And the idea was just to clamp the trap and to monitor the force as a function of time when you fix the trap geometry. And in this case, exactly, you can write what is the force, which is the derivative of the elastic energy. And again, it's easy to show that the change of force in this case are completely proportional to the change of the active curvature, intrinsic curvature. So you are not poised, let's say, by the shell geometry in this case. All right. And so again, you see the rise of force, which is about a second. And again, when you do, you plot data, what you see is here the closure time scale for the intact trap, which is about 1/10 of a second. And this is the closure time scale for the cut trap and the clamp trap. And you see that you have about the same value of the closure time, which is about few seconds here. All right, so this is the active part. So now, yeah. No, this is the time of the exponential relaxation. So you fit the dynamics by an exponential, and you have a time scale. That's how it is defined experimentally. Yeah, even for the intact trap. So how you fit, because for the intact trap, the shape is not fully exponential. The way we did it is that we compute the time of maximal velocity as the origin of time to make the fit after. Yeah, this is the way, and this is kind of arbitrary, but it gives the second part of the time scale in the. Yeah, it depends, because if for instance some traps that are abnormally curved, can take a very long time before reaching the instability point and snapping quickly. And in this case, you have I don't know if I have not the dynamics of an intact one, but in this case, this pre-time can be very long. Yeah. So here, this is how the time is defined. And so then the question we asked was, okay, what is the motors for this active bending? And I told you that in plants, most of the time people argue that motion comes from water flow that induce change of volume of the tissues. And when you have bending, usually this is associated to shrinkage on the inner side, for instance here, and then expansion of the outer side, so differential expansion. So one possibility, and that has been raised by many people in the field, is that this active motion is an osmotic flow from one side of the trap to the other. And this is what we wanted to test. So this is not easy to measure the flow inside, but what we have tested is estimate the time it takes for water to move from one side to the other by two different ways. So the first ways, and I'm not going to detail too much if I want to have time to talk about the mimosa, but the first technique is to use what people call a cell pressure probe, which is you put a needle in the cell, connect to a pressure sensor inside a completely closed microfluidic chamber, and there is here in the chamber a mobile piston, so you can suddenly perturb the cell, adding some extra water in it. And because the cell is permeable, this water pressure will relax toward the initial equilibrium. And this time scale of relaxation is precisely the time it takes for water to go from one cell to the other. So that's what I call a single cell water exchange time scale. And this is something, there are a lot of dispersion, but this is within a second. So one cell takes a second to exchange water with the neighbors. So that's one technique. And then by a very simple, very easy model, like a chain of cell, like people are doing a chain of a spring, for instance, or whatever, you end up with a diffusion equation for the water transport. And from this diffusion equation, you can estimate that the time scale it takes to cross n cell is simply n to the square times the time scale for one cell. Okay, so this is the basic of poroelasticity in this case. All right? And when you do this, you estimate the time scale to about a minute, because there are several layers of cell to cross. Okay, so that's too long. And the other method we use is maybe more more straightforward. The idea was just to measure the swelling of piece of tissues and to deduce the diffusion coefficient of water in the tissues, and then to estimate from the diffusion equation the time it would take to cross a given thickness of the leaves. And again, when you do this, so you put tissues in sugar and then suddenly in a less concentrated sugar. So you track the motion of the tissues, which is due to the swelling of the tissues. And so you end up with a diffusion coefficient, and from the size and the standard relationship between size and diffusion coefficient and time scale, you end up again with another estimation. And this is summed up here. Those are the estimation of the water transport time scale across the trap. And it seems, although it's highly dispersed, and so we are pretty cautious about it, but it seems that it's above the time scale we measured for the active motion. Okay, so there is another thing. It's not water flow transport across the trap. And so the second main hypothesis we have tested with Jong-Gun is that, okay, and this was proposed by Hodick and Sievers, is that in order to achieve active bending, another possibility is to use a kind of sandwich materials represented here. Most of the plants actually are composed of two very different kinds of tissues, the epidermis here in green and the central tissues here in blue. You see the central tissues and the epidermis. And most of the time, the central tissues are in compression. They want to be longer, like the core of a carrot, of a rhubarb, or whatever. If you cut in two, it tends to split like that. Okay, so they are in compression, and they are not expanding because the two epidermis are pretty stiff and prevent the expansion. And so you immediately realize how you can create bending. If you soften one part of your epidermis that becomes softer, then you release some of this compression energy and you have bending like that. Okay, so it's more local mechanisms than mechanisms that needs to transport water from one side to the other. All right, it's like, it's again a kind of a release of elastic energy. But on the tissue level, not on the wall trap level in this case. So we try to test this, and Jongen, because it was a proposal, but there was no in situ evidence of this. So what she used was, she used nano-indentation, so we have this in the lab, to poke the cell of the epidermis, both on the inner face and outer face, using standard, back and forth cycles of indentation like that. It's a bit difficult and tricky to maintain the trap, but you do this on living trap. So you poke it before the trigger, and then you trigger the trap, and then you poke it again, and you measure if there is a difference of stiffness. And actually, there are differences of stiffness. Here, this is the inner face, and this is the indentation as a function of force, as a function of indentation depth. So you have the classical cycle because you have small viscoelasticity. And you see that there is almost no difference before and after triggering the trap. Although outside, when you compare the light green curve here before triggering to the dark green curve after triggering, it seems that the medium becomes softer indeed. And you can repeat this on many traps. So there is a kind of an asymmetric change of mechanical properties that occurs when you trigger the trap. So there is something in mechanics, right? And so we are pretty happy to see this first, and then you can repeat this, and this is systematic measurements on many traps. As a function of time, what was nice in these measurements is that Jongen managed to make measurements over a long time scale before in order to be sure to have a reference value. And so here in red, pink, it's the internal phase. Here in green, it's the external phase. And what you see is that indeed, when you trigger, there is this drop of stiffness of the cell of the outer layer. And this does not occur for the internal phase. And this is maintained. It's not a transient decrease of stiffness. And this is maintained at least an hour during the measurements. Then we struggle sometimes because we wanted to have a real-time measurements during triggering. So we wanted to resolve the time scale of the change of stiffness. And we cannot use that techniques because the silicone paste here to fix the plants was too soft. There was still residual motion of the reference plane, so we cannot make measurements during the indentation. We cannot indent during the motion. We had to indent before and we had to indent after. All right. This is the questions we are asking after. Yeah, this is really this. Yeah, but just to say that, yeah, we could not manage to have measured faster than 2 minutes post triggering. So the idea with Morgan and Marie, there were two interns. We completely changed the setup in order to really try to fix completely the external phase and maintaining a very, very small windows for the tip of, it's not an AFM, so it's a nano-indenter, but still we did this. And also what we did is higher frequency of the indentation force around a mean force in order to try to keep as much as possible always contact to the plants, all right? Although there is always a little motions, but you see now we have modulation of the indentation force around the mean. And here you see this is the modulation of the indentation depth. And look at the figures. Here this is 20 nanometers. So now we are really poking, oscillating around a mean depth very gently. And also, in order to have no perturbation, we trigger the trap using a laser, so we have not to touch the hair, and which also puts vibration to the force measurements at that, because of this very small scale measurements, small level of force and indentation. And when we did this, we had a lot of failure. So many traps just did not respond because they were too constrained or whatever. But for few traps, we could manage to see indeed a drop within few seconds. So it seems that really the stiffness decreased that fast. I mean, on the time scale of this active motion. All right, and I just check the time because, and then came the questions that Drazen asked. Is that all right? I told you that a plant cells looks like a balloon, an inflated balloon. But so if you poke a balloon and it becomes softer, it's not easy to know whether it becomes softer because the pressure inside decreases, or whether because the elastic, let's say the Young modulus of the elastic wall decrease. It's not easy at all because you see the scale, and this is the real scale of the indenter, which is a few microns width of the tip of the cone sphere of the nano indenter, and the typical size. This is a electron microscopy image. The typical size of the cell. So, and the cell wall is pretty thick. So it's not easy to use asymptotic expression of indentation of a very thin balloon or stuff like that. It's not easy to disentangle that. But what we use is actually a more qualitative but discriminating way than the force signals of the indentation, is to say that, okay, actually the response of the shape of the epidermis should be completely opposite whether this is the pressure that decreases or whether this is the elastic modulus of the cell wall that decreases. Because in one case, you can imagine this is a camp mattress, when you inflate a camp mattress. If you deflate the camp mattress, what you expect is that the bulginess of your mattress should decrease, okay? Whereas, it's not easy to do, of course, but if the Young modulus of your mattress would decrease, then the bulginess for a given pressure would increase, okay? So that's an opposite response to our questions. So what we did is a kind of print, digital print image of the trap. So we mold, again with a silicone paste, we mold the trap before triggering and after triggering. And each time, so we remove the trap, the mold, and then we scan it, all right? Here we use a chromatic confocal, how you call it, profilometer. And then you have the profile of your surface, like that. And so you can compute the RMS of your surface before and after triggering. And when you do this, again, it's very distributed, but systematically, you see that the change of bulginess, so the change of RMS divided by the initial RMS, is shifted to the positive, which means that we are in this situation where the stiffness of the cell decreases because actually it's the cell wall Young modulus that decreases, not the turgor pressure, which is a bit surprising. So since I want to talk, have sometimes to talk about the mimosa pudica, I just want for the next 3 slides just to say that we have tried to be more quantitative and try to infer the change of Young modulus associated to this observation. And here we use 2 things. We use our experimental input, which is what is the shape of the epidermis before and after, what is the indentation of the cell before and after. So that's 4 experimental inputs. And we use this and COMSOL, so a very basic simulation of a cell having the same geometry that we've measured using the electronic microscopy. So we have, and then we use a kind of iterative algorithms, which means that knowing the shape before and after, and knowing the indentation curve before and after, we end up with two two different values of the change of Young modulus. And we have one unknown, which is the natural shape of the epidermis when it's completely out of Tugger. And we loop our simulation in order to for these two values to match. And this gives us the Tugger pressure inside the cell, the initial Young modulus of the cell wall, and the final Young modulus of the cell wall. And it gives about almost 40% of drop of the Young modulus of the cell wall. Okay, so that's it for the venous flytrap. I mean, we can put this in a very simple model and see that at least this change of Young modulus seems to be consistent with this very simple sketch I've just shown you, where it's trivial to compute the change of curvature associated to a change of Young modulus, knowing the thickness of the trap. Okay, so a very basic scaling argument like that predicts some bending deformation, and the bending deformation predict is within the order of magnitude. Of course, it's not at all an exact value, but it's within the order of magnitude of the change of both bending deformation and change of natural curvature that we measured experimentally. So at least this is not completely off in terms of order of magnitude, that change of Young modulus is needed in order to generate this active bending. But the origin of the intrinsic origin of this molecular change, of this cell wall change, really we don't know for the moment. We've seen that there are some calcium waves, but we suspect that this is not at all the calcium that directly softens the cell wall, although it is known that calcium can affect the mechanical properties of the plant cell wall, but we have some results showing that this is not the case. So people in the cell wall community most thinks about change of pH that can rapidly modify the link between the microcellulose fibrils inside this hydrogel matrix, and that could change the mechanical strength of the cell wall rapidly. But that's something a bit still completely. So it's more like a slippage of a slippage. So there are some link, and it is known that changing the pH activates some enzyme that are pH dependent, enzyme that modify the strength of the link. That's a good point, because for us it's really the Young modulus that we measure. It doesn't seem that the plastic part has changed that much. So really, I don't know. I don't know for the moment. Yeah, whether what we have measured is about the Young modulus, so reversal part. Okay, so that's kind of a first strategy the plants have used to produce rapid active bending. So there is no elastic instability here. It's not just a fast local change of cell wall properties. So we're gonna meet this plant now that Mathieu is working on, so the Mimosa pudica, to see how does it work in this case. And we have seen that it's a different strategy. So again, I can show again the movie, and here too, the same Japanese team. Actually, they are the only. This team they are the only one in the world to be able to genetically modify those kind of plants to visualize some molecular signals inside the plants. It's not at all, of course, usual plants, but they have managed to observe the same kind of calcium signals. So you see, there is a bright signals that propagate all over the plants. And where are the locations of the motion in these plants? Actually, the motor organ of the Mimosa pudica, they are called the pulvinus, and many plants have pulvini. And on these leaves, composed leaf, there are three levels of pulvinus. The main here, the primary pulvinus at this junction, but then there are secondary pulvinus here, and then tertiary pulvinus, the smallest at the intersection of the little leaves here. And Mathieu is studying mostly this one, so the main pulvinus. And this is a close-up, so you see it's a kind of a very thick and short rod of 3 millimeter long and 1 millimeter about thick. So very short and and thick rod. And this is a real-time movie where someone is poking the plants, and you see the nice bending of that rod. And what is amazing here is that if you compute the deformation of that rod, it's about 20 to 30%. So it's 10 times more than before. It's a huge deformation because it's very thick. You see, it's a big curvature and very thick. No, the poke, there are some mechanosensitive cells that are not the hairs here. Here the hairs are, you can remove it. It's completely irrelevant for the motion itself. So they are not sensitive. And if you make a cross-section of that pulvinus, you can see that composed of tens of thousands of cells packed, densely packed here, right? So when you open a textbook, how this is explained usually? Well, there have been many studies showing that indeed when you trigger the trap, there is a flux of ion very rapid from the inside of the cell of the lower pulvinus, so which is the pink region here. So some ions rush out the cell, and what people believe is that, okay, it is associated to a very rapid drop of turgor pressure by osmosis, and then to a very rapid water flow that deflate the lower part and inflate the upper part. Okay, so it's really a water transport, how it is explained, and it seems to be there are some solid evidence, at least from the electrophysiological point of view, that indeed some rapid flux of ion are involved in this motion. But on the mechanics part, there have been very few studies actually. So most of the studies focused on the electrophysiology. So what we have done with Mathieu is to try to make two things. First, we say, okay, this is a nice, there is no movie here? I thought, yeah. This is a very clean motion because it's also reversible. So within 10 minutes, the plant resets and you can trigger it again. So it's fully reversible deformation. And it reminds us a kind of a muscle. So we wanted to know, okay, what's the characteristic of this muscle first from a purely macroscopic point of view? And then how this motion can sustain the idea that this is a rapid water flow due to osmosis. Can we believe in this, and can we test this again? So a bit the same as the first plans. And so briefly, what we have done is really to mimic what people have done with muscles long time ago, where they measure the power of a muscle as a function of the speed of contraction, for instance. This is called, for instance, the Hill's law, which is simply that you apply a force to your muscle and you measure what's the maximum speed, for instance, the muscle can exert and so on. And there are some laws saying that, okay, when the muscle is load-free, this is the fastest contraction it can have. And of course, there is a maximum load where the muscle cannot contract anymore. So you have a kind of a hyperbolic curve like that. And from that, you can compute the power, F times V, of the muscle as a function of the force or as a function of the velocity. So here, it's not really a kind of a contraction. It's more like a torque and an angular speed that we have tried to measure. So it's purely macroscopic motion. We try to build these motors. I mean, what is the characteristic torque versus angular velocity of this muscle? And for that, we need two things. We need a speed and we need a force. So the idea was just to measure accurately. So again, I don't know if yeah. So here, this is the motion triggered by two electrodes. So you can very nicely measure the angle as a function of time. And here, this is on a test machine. You just blocked the plant and you measure the torque as a function of time. And making a very simple mechanical model, which is shown here, you can, from these two quantities, which is the free motion on one side and the blocked motion on the other side, you can, from this a pair of measurements, plus this model, you can extract what I call the Hill's law. So the Hill's law for this green muscle, which is the torque as a function of the angular velocity. And this is shown here. And what you see is that actually there is mainly a decreasing part. So I'm not going to comment now this part here, but you see a kind of almost a linear decrease of the torque as a function of the speed, right? And from that, you can also compute the power of this little pulvinus. And what is funny is that if you compute the power density, so watt per kilogram, you end up with figures that is pretty close to what people have measured for animal muscles. So although the mechanisms, the microscopic mechanisms is completely different, here we believe it's driven by osmosis, where here it's driven by the sliding of actom-using motors and the burning of ATP. The figures are not that different. Don't know if it's a pure coincidence, but maybe not. And then yeah, but I want, yeah.
It's also more efficient from, let's say, like cell energetical.
Yeah, I don't know. I've not made this budget at all. And I won't stick too much on this 2.5. Just to say that this is the same order of magnitude, because there are big dispersion of all these things I'm showing you. I mean, it's this is for one pulvinus, and then you have to average the value over many different pulvinus. So, but I've not made the budget from the let's say the cost of the osmotic change of the chemical potential of the water, for instance. What you need to move for ions, for instance, not made this. That's interesting, yeah. And okay, so just to say that, okay, so we have the macroscopic, let's say, motors, which is torque versus angular velocity, and we have the unit microscopic osmotic motors, which is this cell with a semi-permeable membrane, for which there is a very famous law between the water flux and the change of chemical potential of water, which is a standard thermodynamics relation between the flux and the gradient of thermodynamic potentials. Here there is the membrane permeability, all right? And so you have a similar characteristic. So here at the microscopic level, you have a characteristic like this between the pressure and the flux. It looks a bit the same. So you can say, okay, can I build a very minimal model going from this to this? And it's pretty easy. You just have to assume that, okay, you have a very simple geometry, and all the cells are pushing together. And when they swell, you change the length of one side and the other side. And then you can link the macroscopic torque to the microscopic change of osmotic pressure. You can also link the maximal velocity of this Hill's law to both the change of the osmotic pressure and the membrane permeability. So you can have this link that involves here the number of cells membrane you have to cross to go from one side to the other side. All right? So you can say, okay, now that I know this, I measure the two quantities here. For instance, I measure the torque and I deduce the change of osmotic pressure. And what you see is that it tells you that the cells have to change its osmotic pressure by about 5 bars, which is typical for plant cells. So nothing is very special in terms of stress or in terms of force exerted by this muscle. I mean, this is typical of plant cells, few bars in the cell. What is much more surprising, of course, is the speed, because here, if you extract a cell permeability from the maximum speed of our pulvinus, then you find cell permeabilities that are way too large compared to what is known in plants or even in animal cells. I mean, there are at least 2 orders of magnitude difference. So clearly, there is a problem here. I mean, you cannot account even because of the simple model by these 2 orders of magnitudes. And more than that, you can also do the same kind of experiments I've done with the Venus flytrap. You can take a piece of tissues, put in sugar, and then suddenly, so you expel water slowly, you reach equilibrium, and then suddenly you transfer this in a pure bath of pure water. And you look the time scale of swelling of your tissue. So again, it's a kind of proelastic time scale, and here what you see is that it takes minutes. Even if you take a tissue that has the same size as the thickness here of the plant. So it cannot be that an osmotic flow like that can drive this motion. So we are very puzzled since there have been many pretty robust evidence that osmotic flows is involved in this situation. And so we had some, we were really wondering, is there any water movement in this case? Is it like the Venus flytrap and so on? But then we looked closer to the movie like that. And I'm going to run this movie again. The movie is bigger, and maybe you will notice something here when the Venus is moving. So do you notice that that there is a rapid change of color? Or no? And I do it again. You notice this? So initially we had tens of movies like that and saying that, okay, it's just a kind of a shadow that just move like that. So Mathieu blocked the stem in order to, because we moved the light and so on, but always we like, do you see the shadow? Yeah, I see the shadow. Is it a shadow? And then he blocked it. Not sure you're going to see, but now it is blocked, and we are no longer poking the trap to trigger it, but we use the electrode techniques, so it's contactless. You are not. And now I run it in loop, and it's much nicer now. You see that, so it's no longer a shadow. Okay, there is no, and you can measure the light reflected, and there is a clear darkening that occurs over the time scale of the motion. That's for sure. And when you see this, you immediately, when you are doing granular materials, of course, you remind this kind of stuff that we all see that when you wet something, it becomes darker than when it's not wetted, like a t-shirt, anything, snow, sand, whatever is white. As soon as you put water on it, it becomes darker. All right, and it becomes darker for what reason? Very simply because before there are a lot of air between the grains, and so the optical index mismatch between the silica grains and the air is large, and so there are a lot of backscattering going to your eyes. So it's bright here. Okay, this is multiple scattering. Here, if you have now water between the grains, the mismatch of optical index is less, and so a ray of light going here has less chance to come back to your eyes before being absorbed by the medium. So it's darker. And to check this, we did the opposite experiments, so studying transmitted light in this case, and it's pretty clear, less clear, but so it's again in loop, very less clear with the screen, but it becomes lighter, brighter in this case. Okay? Instead of darker, when the light is coming through the sample and going to you, then it's brighter. So it's really what you expect from multiple scattering. If there is a scattered medium that suddenly change the optical index mismatch between the scattered and the external medium. So the idea was, okay, maybe what we see is really here water motion, just using this reflection of light. And so the hypothesis was, okay, in the Venus there should exist some interconnected network of channel between the cells that initially are filled with gas or air or whatever. Okay, so that's the hypothesis, and upon triggering those air channels, they are rapidly filled by the water expelled from the cell. All right, thus making the backscattered light decrease and the transmitted light increase, and then this interconnected channel network could be a way for the water to move very fast without the need to cross all the membranes of the tissues. So that's a pretty basic idea, actually. So we wanted to test this differently than just by light scattering, more directly. And so we went to Soleil last year in April to try to make in situ experiments microtomography of the pulvinus before and after triggering the pulvinus. But we need a high resolution and very fast tomography. So in Marseille, we have some tomograph in the lab, but it's okay for pretty static stuff, but not at all for that kind of... And also, we don't want to kill the plants. So the nice things with synchrotron is that it's more like it's a very coherent beam. So you can use the phase, the interference of the beam to enhance the contrast, not only the beam absorption. So this is the setup. We trigger externally electrically the trap. And indeed, when you look at the micro CT scan, you see a lot of air pockets between the cell. And you see a strong gradient of air pocket density also. So it's really like a packed emulsion, let's say, with air between. Or another image, it's kind of a foam, but you reverse the gas and the liquid. So it's an inverse foam, let's say. And in a foam, the plateau borders, they are all connected. And here, you can reconstruct the kind of plateau borders of this pile of cell, which is a kind of a place where water can flow without crossing membrane. And also, we can also look before and after. Indeed, after triggering, we can see in many cases that these channels becomes filled with water. Okay, so it seems that really the mechanisms of using this kind of I don't know how to call it. It's not a conducting vessels like blood or like for trees, very organized. It's more a random network of conducting tiny micrometer size vessels. All right. And to finish, the last hypothesis we try to test is that indeed, is it true that thanks to this interconnected network of channels, is it true that water can flow very fast and swell the tissues within a second? And to do this, we have, because you remember that before we put some tissues in sugar and we swell the tissue using a gradient of concentration. So it's an osmotic forcing. Here, to test this hypothesis, we need not an osmotic forces, not a gradient of concentration, but really a gradient of hydrostatic pressure. We need really to push water flow with a pressure inside the tissue. And the trick we use is just evaporation. So we slightly evaporate the tissue. So now the pressure inside the tissue is in tension. And we suddenly put this desiccated tissue inside pure water, so it absorbs water again. And now it's a pressure-driven flow, capillary pressure-driven flow. All right, so it's a different way to swell the tissues. And when you do this, so this is the dry tissue that has been cut and let it dry for a few minutes, and suddenly it is flooded by water. And this is in real time, and you see then that within seconds you can see a change of curvature. So really it means that those interconnected channels are highly wettable and really can make water to go very, very fast in the tissues. So that's just a quantitative movie to see. It's like a sugar. It's like you put, it looks like this, except that then there are a lot of cells that swell. So it's not like a rigid matrix where you have a capillary rising. Here it's more like all the cells absorb water by osmosis, but through a capillary network. And yeah, the time scale is very short. So it's still in progress, all that measurements. And I just wanted to finish because it's very late. I'm very late. And I just want to say that, okay, just to say that people usually say that active actuation is limited by water transport because it has to cross membrane and so on. And just to say that those two plants seems to have found two different strategies to bypass the membrane and to enable long distance transport flow without this osmotic barrier all the time. Okay, by two different ways, actuating the cell wall, the mechanics of the cell wall, or having these architectures of the networks that transport water very fast. Okay, and thank you very much. And this is all the people that I've worked with.
Merci, thank you very much.
Where the water comes from?
So for the Mimosa pudica,
Yes.
so for the Mimosa pudica, it comes from the cell. So all the cells lose their water and transfer within the pulvinus. They lose.
It seems to happen because after darkening, your pulvinus is inflated.
No, it decreased on that part. On that part, during the
But your movie shows
yeah, for this movie you mean?
the one.
This one.
The first movie where you show the darkening of the pulvinus
Yeah,
made by Matthew.
yeah, this one. This one or this one?
This one.
Yeah. No, the darkening. No, the darkening is everywhere, but it starts. You see, it starts underneath.
Yes. But, okay. But there is another video where you prevent
Yeah.
the bending.
So this is this one.
No. There is another one maybe. Okay, I saw one where it seems that pulvinus, without changing the lower geometry, like inflates a little bit.
Yeah, but actually, whether it inflates, so the part that deflates is the bottom part, the part that inflates is the upper part. But the change of color is not linked to, I would say, it's not linked to whether it deflates or inflates. It just means that before, around the cell, the channels were filled with gas, and after, there are water that flood the channels. So the darkening can be is there both in the deflating part and the inflating part. So the gas, that's a very good point. So actually, maybe you have noticed that there are a lot of big pockets close to the around the conducting vessel here. So actually, the system is full of gas and a large quantity of gas. So this large quantity of gas is highly compressible. And actually, where the gas is coming from, so it's compressed, and you can see this in this movie. Maybe you will notice, because now the system is open, maybe you will notice that here there are many bubbles. I don't know if it's very easy, but there are many bubbles that are expelled because now you have cut it. So here the water rush inside the network and push the gas bubble that goes outside here. Yeah, but it's a huge quantity of gas and it's highly compressible, the gas.
The whole movement is reversible. Does it come back after
Yes,
what?
after 10 minutes, about 10-15 minutes, yeah. And the darkening reversed in this place. Yeah.
Yes, thanks for the presentation. I have a question on the Venus flytrap. So you measure a change of Young's modulus. Is it possible that this is due to a change in anisotropy of the membrane of the Venus flytrap?
I tend more to think of a change of either the link between the microfibrils or the change of the matrix between the microfibrils, because so a changing of anisotropy is pretty difficult, because the cell wall, it's true that it's made of so it's a kind of isotropic matrix and with a lot of microfibrils of cellulose that are very stiff. And the cell is sometimes isotropic or anisotropic depending on the orientation of the microfibrils, but this anisotropy is controlled upstream, really upstream, when the microfibrils are synthesized, and this is a very long process. And so I have difficulties to see how those orientation can change within a second. I mean, this is you see.
Yeah. I mean, in man-made systems, you have like polymer liquid crystals, for example.
Yeah,
can do
exactly.
this.
Yeah, but then it's liquid crystal. So here, this is within a very elastic and solid matrix.
Okay.
Yeah, around this is pectin. It's a gel itself. So yeah, rigid gel.
Thank you. About Venus flytrap, you measured once the turgor pressure with your needle and
Yeah, yeah,
and would it be possible to measure the turgor pressure during
yeah. We tried
and
this
to
for a long
see-
time.
it's-
Yeah, it was actually one of one of the goal of. I put the name Mathieu, but not Mathieu Rivière, Mathieu Colombani. During his PhD, he spent a lot of time to try this, and in the end, he managed to do only measurements on a fixed trap. I mean, not during motion. I mean, this cell pressure probe measurements are very difficult and prone to any leakage if there is some motion, relative motion between the tip and the cell. And then, yeah, we tried, but we never succeed.
Thank you very much for a fantastic talk, in my opinion. I seem to remember that calcium is a very good use as a marker for electricity propagation in muscles
Yes,
in animals.
absolutely.
So is it the same thing in plants, actually? Is it used as a marker for the same reason?
No, so electricity is not very studied in plant, much less than actually calcium seems to be, I'm not expert at all, but it seems to be a marker of many things in stress. At least stress, it's a kind of a stress marker. As soon as you have a stress in plants, it can be a cold, it can be
Okay,
dry or
so
whatever.
here it's more a
It-
stress than
yeah,
electricity
But
signal.
people do not very clearly understand. It seems that calcium comes from the outside of the cell, but also from the inside, like noyau. And depending on where it comes from, depending on the spatial place, it can give different response as well. But yeah, for the electricity, it's a topic that is starting. So
Okay.
people are starting to use calcium as a proxy for that. Yes. Yeah.
Thank you.
Super, merci
There's the
beaucoup.
last one, but
Ah.
I don't know.
Oh,
It is very.
These snap mechanisms here occur with the treatment,
Yeah.
but there are some snaps that happen without
Yes.
passiflora. It's snaps open in smooth condition. Can you say?
Yeah, so apparently I've never seen the Venus flytrap closed by accident, but this is something that I've been observed and studied quite a lot for this system by the group Philippe Marmottan in Grenoble. So this is an underwater plant, that kind of a pocket where there is that expel water a little bit so that the pressure inside the pocket decrease, so it's a bit the depressure, and there is a door, and the door has the shape of a dome. So it's really like a shell, and actually it seems that in this case, since the plant keeps expelling water, the pressure keeps decreasing, and sometimes, boom, spontaneously, so it's not a trigger, it's more like spontaneous, so you reach the buckling threshold, I would say, and so you can see spontaneous and many buckling like that in nature. Yeah, but still there is a parameter that is smoothly varying and the pressure. It's not, I mean, yeah. But for sure, the Venus flytrap is not is in a metastable state. It's not like sometimes you have these toys, you reverse it and you let it and it will snap. So it's not a strictly metastable state. This one is broken, so it's a metastable state. But otherwise, so I need something to snap it. But so the Venus flytrap, it's a local stable state in the open states for sure. Yeah.
Merci encore.